In this video, we will talk about Cri-du-Chat syndrome, Wolf-Hirschhorn syndrome, and Williams-Beuren syndrome. But before discussing these syndromes in detail, let's talk about the several generalizations of chromosomal syndromes, which will help you in learning these chromosomal syndromes. Although chromosomal syndromes are typically quite variable, several generalizations are nevertheless possible. So, most chromosomal abnormalities are associated with developmental delay in childhood and intellectual handicap in teenagers and adults. Most produce characteristic facial appearances. Most are associated with retarded growth, and many include congenital malformation of the heart. Well, it is important that every child with multiple congenital abnormalities should be examined for chromosome damage at an early stage. If a condition is suspected, antenatal diagnosis is generally possible by chorionic villus sampling at 10 to 12 weeks or amniocentesis at 16 weeks. Let's understand chorionic villus sampling. In chorionic villus sampling, a syncytial trophoblast biopsy is aspirated via catheter through the cervix or by transabdominal puncture in chorion frondosome at 10 to 12 weeks gestation, both guided by ultrasonography. The chorion frondosome is that part of the conceptus that forms as the villi grow larger on the side of the chorionic shell next to the uterine wall. The discus-shaped placenta develops from the chorion frondosome and the decidua basilis. The early timing allows diagnosis by about 12 weeks. But there is an associated risk 0.5 to 2% above the spontaneous abortion rate of 7%. Since syncytial trophoblast nuclei divide rapidly, karyotyping is possible without culturing. But cultured material provides more reliable results. DNA can be isolated directly from chorionic villi for molecular testing. The syncytial trophoblast is the placental barrier between maternal and fetal blood that allows exchanges in nutrients and gases, and also represents the endocrine tissue of the human placenta. Next is amniocentesis. Amniocentesis is especially valuable at 16 to 18 weeks for estimating alpha-fetoprotein concentration and acetylcholinesterase activity in pregnancies, or for analysis of chromosomes, DNA, or enzyme activities in cultured cells. After prior localization of the placenta by ultrasound, a needle is inserted aseptically through the mother's abdominal wall and into the amniotic cavity, and a 10 to 20 milliliters sample is withdrawn. This carries a 0.5% risk of causing miscarriage, in addition to the natural risk of 2.5% at 16 weeks, or 7%, when alpha-fetoprotein levels are high. Let's discuss amniocentesis versus chorionic villi sampling. Well, amniocentesis involves the extraction of a small amount of amniotic fluid that contains fetal cells, whereas in chorionic villi sampling, suction tube is used to remove fetal cells from chorion where placenta will develop. Now let's discuss deletions and segmental aneuploidies. First, microdeletion syndrome is Cri-du-Chat syndrome. Genetics of Cri-du-Chat syndrome is 46XX delete 5p15.2. Means there is deletion on chromosome 5 at arm P of band 15.2. Frequency of Cri-du-Chat syndrome is 1 out of 20,000 to 1 out of 50,000. Possibly 1% of profoundly learning disabled. Lifespan of Cri-du-Chat syndrome is. Survival into adulthood sometimes occurs. Management of Cri-du-Chat syndrome is that a structured exercise regime is recommended, plus verbal stimulation and correction of squints. Now let's discuss features of Cri-du-Chat syndrome. The newborn baby has a low birth weight, and Cri-du-Chat is a French word which means cry of the cat. So, patient makes a distinctive cry similar to that of a kitten due to underdevelopment of the larynx. Other features are small head, widely spaced eyes, epicanthal folds. An epicanthal fold is a skin fold of the upper eyelid covering the inner corner of the eye. Depressed nasal bridge, down-turned mouth. Patent ductus arteriosus is present in 30%. Patent ductus arteriosus is a heart defect that can develop soon after birth. It affects the way blood flows through a baby's lungs. Mild patent ductus arteriosus might not need treatment, but some children with the defect may require catheterization or surgery. The face is round in children, elongated in adults. Severe intellectual handicap. IQ level is nearly 35. You can memorize important points about Cri-du-Chat syndrome by keeping in mind that a baby is sitting, holding digit 5 in hand, and a crying cat sitting on her lap, holding digit 5, because there is mutation of chromosome 5, and cat sitting on her lap, because Cri-du-Chat is a French word, which means cry of the cat. So, patient makes a distinctive cry similar to that of a kitten due to underdevelopment of the larynx. Moving towards Wolf-Hirschhorn syndrome. Frequency of Wolf-Hirschhorn syndrome is 1 out of 50,000. Male to female ratio is 3:4. Genetics of Wolf-Hirschhorn syndrome is, for example, 46XY deletion 4p16.1, which means there is deletion on chromosome 4 at arm P of band 16.1 and balanced translocation in 10%. Management of Wolf-Hirschhorn syndrome is that developmental checks are necessary. There is failure to thrive, but lifespan can be into the teens. Now let's discuss features of Wolf-Hirschhorn syndrome. Microcephaly. Microcephaly is a condition where a baby's head is much smaller than expected. During pregnancy, a baby's head grows because the baby's brain grows. Microcephaly can occur because a baby's brain has not developed properly during pregnancy or has stopped growing after birth, which results in a smaller head size. Hypertelorism. The term hypertelorism means an increased distance between two body parts. It usually applies for the eyes and termed it as ocular hypertelorism. Epicanthic folds. An epicanthal fold is a skin fold of the upper eyelid covering the inner corner of the eye. Lack of indentation of the nasal bridge is described as resembling the protective nosepiece. Cleft lip with, or without cleft palate. Low set ears, short upper lip, hypotonic at birth, and muscle tone remains poor. Heart defects, convulsions, and undescended testes, and severe learning difficulties. You can memorize important points about Wolf-Hirschhorn syndrome and cat's cry syndrome, such that Wolf-Hirschhorn syndrome is 4p deletion, and cat's cry syndrome is 5p deletion. Now let's discuss Williams-Beuren syndrome. Frequency of Williams-Beuren syndrome is 1 out of 10,000. Genetics of Williams-Beuren syndrome is that it has 1.5 megabases microdeletion, possibly inherited as autosomal dominant inheritance. Autosomal dominant inheritance is a way a genetic trait or condition can be passed down from parent to child. One copy of a mutated gene from one parent can cause the genetic condition. A child who has a parent with the mutated gene has a 50% chance of inheriting that mutated gene. It is deletion 7q11.23, which means there is deletion on chromosome 7 at arm Q of band 11.23. Management of Williams-Beuren syndrome is dietetic advice, energetic play. Intellectual disability prevents independency. Now, let's discuss features of Williams-Beuren syndrome. Broad forehead, short palpebral fissures. The palpebral fissure is the area between the open eyelids. Starry pattern of irides, which is coloured muscular ring around the pupil of the eye in vertebrate animals, located between the cornea and lens. Tip-tilted nose, with low bridge and outward-facing nostrils. Long philtrum, vertical groove between the base of the nose and the border of the upper lip. Full cheeks, large mouth with full lips. Excessive vomiting. Sleeplessness, slow growth rate. Children have a cocktail party manner. Williams kids have what is known as a cocktail party personality. They are very talkative and have very good linguistic skills. But their speech is disassociated, jumping from one topic to another. They have major problems with spatial processing, with outgoing attitude, hyperactivity, and advanced facility in language, but become withdrawn as adults and sensitive especially to loud noise. There are also mental retardation. Supravalvular aortic stenosis. Supravalvular aortic stenosis is a congenital heart defect that accounts for 8 to 14% of all cases of congenital aortic stenosis. It involves a narrowing of the portion of the aorta located just above the aortic valve, multiple peripheral pulmonary arterial stenosis. Peripheral pulmonary stenosis is a narrowing in one or more of the branches of the pulmonary arteries. Dental malformations. Short stature. With sloping shoulders and visual spatial cognitive deficiency. Visual spatial processing is the ability to tell where objects are in space. That includes your own body parts. It also involves being able to tell how far objects are from you and from each other. People use visual spatial processing skills for many tasks, from tying shoes to reading a map. Let's discuss etiology of Williams-Beuren syndrome. Deletion of a segment of 7q carrying about 20 contiguous genes, including the elastin structural locus. Elastin is expressed in the aortic wall. Its deficiency causing supravalvular aortic stenosis. Limb domain kinase 1 can also be lost. This is a kinase expressed in the brain and likely to be involved in visual spatial cognition. You can memorize features of Williams-Beuren syndrome using a mnemonic. Shame. Supravalvular aortic stenosis. Hypercalcemia. Autosomal dominant. Mental retardation. And elfin faces. This was about Cri-du-Chat syndrome, Wolf-Hirschhorn syndrome, and Williams-Beuren syndrome. In our next video, we will talk about DiGeorge, Smith-Magenis, and Sotos syndrome.
Have you ever heard of video books? sqadia.com is delighted to present video books, medical books that have been tailored into a series of videos. sqadia.com videos are presented in the same order as the book's chapters. Explore now and make your learning easy.